Hello, everyone, and welcome to phyloseminar.org. The current theme is transmission trees, and this is the second talk in the series of 3 talks on that topic. Please use the YouTube live comment box to ask questions or tweet at phyloseminar. Today's speaker is Don Klinkenberg. Dr. Klinkenberg works on infectious disease dynamics at the population level. He did his PhD at the Institute of Animal Science and Health in Lelystad, the Netherlands. He then moved to the Faculty of Veterinary Medicine of Utrecht University, first as a postdoc and then as a lecturer. He is now at the Center of Infectious Disease Dynamics, where his focus is on combining genetic and epidemiological data. Welcome, Don, and thanks for speaking to us today.
Thanks, Eric. And thank you for inviting me to give a lecture on this website. I'm not sure how many of you will have seen Caroline's lecture, but if not, please do see it. It's really good. I enjoyed it. And my talk will be on Phybreak, which is a method to infer phylogenetic and transmission trees, and it's a bit a similar type of method as what Caroline was presenting last time with her method TransPhylo. So I'm Don Klinkenberg. I work at the National Institute for Public Health and the Environment, RIVM in the Netherlands, indeed at the Center of Infectious Disease Control. And parts, so I'm doing one part is outbreak analysis more in general. So I'm also involved now in the current coronavirus outbreak or epidemic. And one part is the use of genomic data. And in that part of my job, I've worked on Phybreak, which is a method and an R package to infer transmission trees from genomic data. It's actually based on earlier work from by Rolf Ipma, who also worked at RIVM just before I came. So there's papers in genetics from him, and based on those methods, I've developed Phybreak. So what I will do today is first tell you a little bit about the type of data and the epidemiological setting that I'm thinking of that are relevant for using Phybreak. I will briefly explain the difference between a phylogenetic and transmission tree. It's also something that Caroline covered last time, but I think it's good to show it because it's really a relevant issue here. Then I will go on and tell you about the model, the Phybreak model, and then a little bit about the way the MCMC sampling is done in the package. And I will give you a brief overview of the functionality of the package, so the workflow, how the data are entered, and what you can do with the output. And then very briefly, I will touch upon some new developments. So for the main part of this talk, I will focus on Phybreak as it is currently on CRAN, and which is also the version that is published in PLOS Computational Biology in 2017. I've worked on some extensions, which I will cover in this new development part at the end, which are implemented and can be found on my GitHub account. So they are already available for use. But because I first want to write and publish before I put it on CRAN. So first, the type of data and the epidemiological setting. So I'm thinking of an outbreak, which is densely sampled. So where, in fact, all cases are observed, sampled, and detected. And usually, if you have an outbreak like that, so for instance, in an intensive care unit or an outbreak of animal diseases like foot and mouth disease, where the unit of the epidemiological unit is not the animal but the farm, then all the cases are detected, and the idea is that you also have a sample and a sequence, pathogen genome sequence for all these cases. So usually you have other epidemiological data like maybe contacts or locations of farms when symptom onset was, or other things like age or sex or medication, drug use. And what what you want with these data is answer questions like what are risk factors for infection or spread? Were there any undetected cases? Was it one outbreak or were there actually multiple introductions? And maybe in this case, in this example of the MRSA outbreak in the neonatal intensive care unit, where there were cases in different wards in the hospital, was there a difference with respect to transmission within the wards and between the wards? Well, this is all epidemiological data, but the additional type of data that we often have in these days is the sequences of pathogen isolates, and that's what this method is about. So the sequences is by whole genome sequencing or sequencing of some of the genes, and the type of data is the single nucleotide polymorphism, so the SNPs. So we assume so there's no recombination or reassortment of things like that. It's just point mutations that will be used in this model. And the idea, of course, very generally, is that similar isolates are probably close in the transmission network, and isolates that are very different are far away in the transmission network. And the aim of the analysis is to infer what the transmission history is. So who infected whom, and when did these transmission events occur? And with this question, Phybreak can help. So I will cover the model that Phybreak uses to answer these questions. I will say something about the MCMC sampling method, and it is an R package. So I will show you a little bit about how the R package works. Okay, a little bit about the phylogenetic tree versus the transmission tree. So if you have genomic data, sequence data from an outbreak, such as this MRSA outbreak in the ICU, the naive approach would be to make a phylogenetic tree. And this is such a phylogenetic tree that was made in the original publication where these data were published. And what you can see here is that ward B is so that you can define clades, and that maybe cases that are in the same ward are more or less clustered. So you see the blue samples clustered, 10 and 12 in the middle. So it tells you a little bit about the history of transmission, but it does not tell you actually who infected whom. And that's because this is a phylogenetic tree is always a bifurcating tree. At the tips are the samples, so the patients that were sampled, and the internal nodes are unobserved the sequences that are the most recent common ancestors of the samples, so of the pathogens, but it's not the most recent common ancestor of a host. So the phylogenetic tree gives you the history of the sampled pathogens, and you have the, for instance, the most recent common ancestor in clade 1 depicted here. But it does not tell you who infected whom. And what we would like is a transmission tree where we see the history, not of the pathogen, but the history of the host, who infected whom. And in a transmission tree, you see who infected whom, and there is an order of generations. And one of the, and what you see is that, so some terminology. So we see here an outbreak in terms of generations. So the index case is generation 0, and every next case is the next generation. And we can speak about the infector, which is the person by whom someone is infected, and the infectees, which is the progeny of a case. And what you see here, and what is really different from a phylogenetic tree, is that cases do not, so in a phylogenetic tree, every node has two descendant lineages, whereas here, every case can have 0, 1, 2, 3, or any number of infectees. So instead of the x-axis in generations, in real life, the x-axis is on time. So what you like is not only who infected whom, but also when this occurred. And yeah, So then very briefly something about when an outbreak becomes a larger epidemic. So in Phybreak, the type of data that we will use is a densely sampled situation where every case is observed. But if you have an outbreak like this and it becomes very large, you have increasing prevalence. There are many, many cases. And if in a situation like this, you sample some of these cases, then if you would use the sequences of these cases to make a phylogenetic tree, so to go from every sequence back in history to the infector and to the infector before that infector, etc., then all the hosts that you observe will have most recent common ancestors of those sequences in hosts that are not sampled themselves. So in that case, you have all these MRCAs and unsampled hosts. So in that case, the phylogenetic tree that you will infer is in fact the same as the transmission history, but there's just a lot of unobserved hosts in between. So if you have a sparsely sampled outbreak or a larger epidemic and you make a phylogenetic tree, it in fact does give you something like a transmission history, only not with all the cases observed. So it's almost never the host plus its infector. Yeah, the phylogenetic tree represents the transmission history. That's just what I told. Whereas here we are dealing with smaller outbreaks, and we really would like to infer who infected whom and when this occurred. So in this case, if we have this outbreak, we have the little green arrows showing when each case infected the next case, and these red dots indicate when the cases were sampled. So you can already immediately see that because the time between infection and sampling varies, that a person that is observed earlier can in fact be the infectee of someone who is sampled later. So the order of observation is not necessarily the same as the order of infection. And the phylogenetic relation between the samples is through so-called phylogenetic mini trees within the host. So what you see here is that if within every host there are small phylogenetic trees, and the tips of those trees are the samples and are the transmission nodes. So a host with, for instance, the index host has 3 secondary cases. So the phylogenetic mini tree within that host has 4 tips in this case. So there's 3 from the onward transmission and 1 from the sample. You see the genetic history, so the relation of the genetic history between all the samples are through all those phylogenetic mini trees, which together form the major phylogenetic tree, which is the genetic history of all the samples. And then what you see, so now I just entered some sequences, and with the yellow dots I indicated where mutations occur. And what you see here is that if we start with a sample with only A's, that you see that if mutations occur within a host, that the SNP that arises from that mutation is not necessarily observed in that host itself or in all the progeny of that host. So for instance, in the index case, the top yellow dot results in mutation of the second nucleotide from A to C, and that is not observed in the index host itself because it occurs on this top lineage, but it is transmitted to the secondary case above, and all 3 top cases in this tree have this mutation, but the other secondary cases do not have that mutation. So what you see that this phylogenetic mini trees, in fact, they describe the within host diversity of the pathogen population. Um, so that all the mutations occurring in hosts are not necessarily observed within that host and not in all the progeny. And by inferring the transmission tree using the genomic data, you should actually take care of all those possibilities. So correct for that. So what Phybreak does is an inference of who infected whom by taking account of this within host diversity and the uncertainty of all those mini trees within the host. And it does so by simultaneously inferring the phylogenetic tree, so it's all these mini trees, and the transmission trees in a single procedure. So as Caroline also said in TransPhylo, it is a sequential analysis where first the phylogenetic tree is made, and after that, based on the phylogenetic tree, transmission trees are derived. But in Phybreak, it's a simultaneous procedure.
Can I just ask one quick question?
Yeah.
So I think you said at the beginning that you're assuming that basically everything is sample, or you have dense sampling?
Yes, I do.
Okay. So you showed an example of an epidemic where sampling is probably going to be sparse, right?
Yes. You mean, I can go. You mean this? This
Yeah.
one?
Yeah.
So here it's sparse. Yeah. But I'm talking about this situation where
Got it.
everyone is sampled.
Thanks.
Yeah. Okay. So then I'm going to explain the model. So this is the very small outbreak. And I will explain the model using this very small outbreak of 4 cases. So in this figure, you see for every case, you see the infection time. That's the gray dot. You see the sampling time, which is the black dot. The transmission events are these blue arrows, and the lineages, the pathogen lineages, are within the hosts, and they are depicted in red. And time goes from left to right. So the first submodel. So the top figure here is now the way I also showed it in the previous slide, and in the bottom figure, so it's not in color but in black and white. But you see, in fact, you see exactly the same transmission tree, but now it's drawn as a phylogenetic tree, where the individual 4 hosts are given a different color or a different gray shade in this case within that phylogenetic tree. So that's the way how Caroline plotted her transmission trees given a particular phylogenetic tree. And here you see that in fact there are just 2 ways to plot exactly the same thing. It's a phylogenetic tree and a transmission tree that are consistent with each other. But I will use the above picture in my slides. So I have the outbreak size that's given, the sampling times, and the sequences. So those will be the data, and the things that will be inferred or will be sampled are the infection times of all the hosts, are the infectors, and is the complete phylogenetic tree P with all the nodes, and the nodes are the tips, are the coalescent nodes. And I also define the transmission events as nodes in the phylogenetic tree. And by doing that, it's easy to cut this large phylogenetic tree for the whole outbreak into small mini trees, because those cut points are at these transmission nodes. So each host harbors an individual mini tree. So the first model is the transmission model. And in fact, the only thing that is assumed is that there is some distribution for the time between subsequent infections. So it's between the time of the infection between hosts in the transmission tree. So there is no model for, no explicit model at least, for an offspring distribution. So implicitly it is, and by not making an assumption, implicitly it's assumed that's a Poisson distribution. But because the outbreak is always over, the mean number of offspring will always be 1. So there's not really a need to have an estimate for a reproduction number or something like that. So in fact, the only thing that is explicitly modeled is this distribution for the time between subsequent infections in the transmission tree. So this is the first part of the likelihood that will be used. So it's a likelihood of the infection times and the infectors, and the likelihood, in fact, it only consists of the density of the gamma distributions of all those generation intervals. And in the package, the shape parameter of this gamma distribution has to be fixed, so it has to be chosen beforehand, but the mean can be estimated. Okay, so the second submodel is the submodel for the sampling procedure. So here again, a gamma distribution is assumed for the sampling interval, which is the interval between infection of a host and sampling of a host. So that's very similar to the generation interval distribution and the transmission model. And again here, so that part of the likelihood is just the likelihood of these sampling intervals given the infection times and the parameters, which is a product of all the densities of the gamma distributions. And also in the package, the shape parameter is kept fixed, so cannot be estimated, but the mean can be estimated in the package. So then the third submodel is the within-host model. And the within-host model is the model that describes these phylogenetic mini-trees. And one of the assumptions that is made is that there is a complete bottleneck of transmission. And that's the assumption that is made in the original paper. So that's also what I will explain here. But that is one of the assumptions that I have lifted later. So in the extensions, I will explain you a little bit about how I changed this to allow for a not complete bottleneck and to have multiple lineages transmitted at infection. But for now, we'll assume a complete bottleneck, which means that the tips in the within-host mini trees are the transmission events and the samples, and that the most recent common ancestor of all those tips is censored at the time of infection because of this complete bottleneck. So it must be after the time of infection. And this within-host mini-tree is modeled with a coalescent process. So we need the number of lineages as a function in time. That's plotted here. So we start with 1 and then 2. There's a short period where there are 3 lineages, and the coalescent rate is the number of lineages choose 2 times 1 divided by the within-host population size. So it's population size of pathogens within the host. And the default model in Phybreak for this within-host population size is a linear growth model. So it's probably not very realistic for how the within-host population actually grows. But given the very short time interval, basically the only thing that is what I've learned is relevant in this updating procedure is the topology of the phylogeny of the tree within the host, and not so much when the exact coalescent nodes occur. And the big advantage of this linear increase is that it starts at 0, which means that the coalescent rate goes to infinity near the time of infection, which guarantees you a complete bottleneck. And later in the MCMC procedure, I will show why that is so convenient. Yeah, so the coalescent likelihood is just is the probability of the phylogenetic tree given the sampling times, the infection times, and all the infectors and the parameters, and it's a product of all the likelihood of all the phylogenetic mini trees within all the individual hosts. So it can be cut into small likelihoods for each of the mini trees. And the within host parameter, this growth rate R, can also be estimated in the package. So then finally, there is a mutation model that gives rise to the sequences given a phylogenetic tree. And for that, just the most simple model is assumed, namely a Duke's Cantor model. So that's just that there's a constant rate, and which is the same for every nucleotide and for every position in the genome. So the likelihood for this part is calculated with Felsenstein's pruning algorithm. So it's a likelihood of the genetic data given the phylogenetic tree and the mutation rate. And the mutation rate mu, the log mu has a uniform prior and is always estimated. It cannot be fixed. So the total model is the product of all likelihoods and prior. So it's the posterior for what we infer are the infection times I, the infectors M, so that's the transmission tree, P, this is the phylogenetic tree, and the parameters given the data, the sampling times, and the sequences. And that can, in a couple of steps, be cut into those four independent, or not independent, but into those four processes. So it's in the bottom line, it's the likelihood for the genetic data given the phylogenetic tree, the likelihood of the phylogenetic tree given the sampling times, the infection times, and the infectors, and then the likelihood of the sampling times given the infection times, and the likelihood of the infection times and the infector. So that's of the transmission tree, and then the prior for all the parameters. So I have the idea that I'm going very quickly. So
it's very clear.
It's okay, good. And then I will say something about the MCMC sampling. So in the package, everything is estimated with the Markov chain Monte Carlo, and the parameters. So the means of the sampling and generation intervals are sampled directly from the conditional posterior because it's a conjugate priors. Because of the conjugate priors, it's possible to directly sample them given the complete transmission of phylogenetic tree, of course. The parameters R, which is the within host growth rate and the mutation rate, are updated through Metropolis Hastings steps. And then the most important part is, of course, the updating of the transmission and phylogenetic trees. And that is done host by host. So every time from the current phylogenetic and transmission tree, a new tree is proposed with a certain proposal density and accepted with a proposal with a probability that is the quotient of the likelihoods and the proposal densities. And that is done on a host by host basis. So I will give an example for this simple transmission tree where I do the updating for host 3. So that's what I call in the paper the focal host for this updating step. So the steps that are taken are as follows. First, this host is removed from the existing transmission tree. And at the same time, all the mini trees so the mini tree in that host is also removed, and the mini tree in the infector is removed. But as you see, the transmission link between the infector, which is host 2, and host 4, that is kept intact, but only the transmission link from 2 to 3 is removed. So then a new infection time for the host 3 is proposed, and a new infector is proposed, which can in fact be the same, of course. It can be, proposal can be number 2 again. But in this example, I will propose number 1. And by proposing number 1 as an infector, that the mini tree of that infector is also removed. So now we are left with a complete transmission tree, but with 3 removed mini trees. So in the next step, the mini trees are proposed. Oh yeah, but first, the sampling or the infection time of this focal host is proposed by using the sampling interval distribution, and the infector is proposed by using the generation interval distribution by putting weights on all the possible infectors. So in this case, the candidate infectors are 1 and 2, but in sampling 1 or 2, those are weighted according to the generation interval distribution. So that's especially in large outbreaks, that hosts that were infected a long time ago are not as likely to be selected. Otherwise, it will become very slow. So then in the next step, the mini trees are proposed in the 3 hosts from, or in all the hosts from which they were removed. And that is done by simulating this within host trees with the coalescent model. And that is why it is so convenient to have within host population size model that starts with a population size of 0, because now all the simulations will always end with a complete bottleneck. So it really makes the procedure much faster. And once we have this new proposed transmission and phylogenetic tree, then the likelihood for the only thing that remains is the likelihood for the genetic likelihood. So those will be compared between the old transmission phylogenetic tree and the new tree.
Let's see, before you move on, is it how hard is this simulation with the is this actually pretty straightforward, the simulation with the coalescent model? I mean, you have to basically, it's sort of like a compartmentalized coalescent where you can't have coalescences between things until
yeah, that's very easy. So you already have the tip times, because those are it's the sampling times and the times of transmission, because they are already known. And then it's just sampling from exponential distributions until, so it's backward sampling of every next coalescent event, and then it's randomly sampling which 2 lineages coalesce at all these times. That's a really very fast procedure.
But don't you have some constraints about like the coalescent times for within the individuals have to, that coalescence has to happen before it gets before it receives the virus from the infector?
Yes, of course. But for instance, so if you look at host 2 now,
yeah,
there in the time between the sampling of host 2 and the transmission to host 4, there is only 1 lineage, so there can be no coalescence. So the coalescent sampling only starts at the time of sampling of host 2, and then there are 2 lineages, and then there is just a fixed rate by which those can coalesce, and and you sample from exponential distribution.
Okay. Cool. Thanks.
Yeah. So by doing this host by host tree update, you always have consistency between the transmission and the phylogenetic trees. So you don't have to take care of that. And you always change the phylogenetic tree, so you don't fix the phylogenetic tree and only sample transmission trees. You change both types of trees. And this is also natural design for more complex transmission tree inference. So such as having more samples per patient, because, well, it just involves more complex within host mini trees, but you can just as easily sample or simulate these within host trees. And also to have wider bottlenecks at transmission. Only then, if you sample if you simulate a within host mini tree and the number of lineages at the time of infection changes, you have to continue simulating in the infector and maybe even further back in time. So that makes it a little bit more complicated, but it's all doable. And those have been implemented in the GitHub version of Phybreak now. And it also makes it, well, that actually doesn't have so much to do with the host by host tree update, but it's also possible to use epidemiological data in the MCMC proposal. For instance, distance. So I have now implemented a distance model where, so in that case, I'm thinking about distances between farms. So farms that are more closely located are more likely to have been involved in a transmission event. And you can use those distances both in the proposal of the infector or only in the evaluation of the likelihood. But by this host by host tree update, you can also use these data already in the proposal by putting larger weights on the more distant or more on the closer candidate infectors. And I want to include also other type of epidemiological data, like the censoring of infection times. If you know when people were cured or when they were sampled and were still negative, or maybe contact time censoring. For instance, if you know when particular hosts were in the same ward or in different wards in a hospital. Those I still have to do. Those are plans that I'm working on. Okay, so then a little bit demonstration about the package. So first of all, there is one function. So I'm now analyzing this MRSA data set in the neonatal intensive care unit. So first of all, there's a function Phybreak data where you can enter all the data. So you have to enter sequences and the sample times, but you can also enter maybe you can also already enter a phylogenetic tree or the infectors to start if you want the procedure to start at a particular phylogenetic tree. And you can, so if there are multiple samples per host, you also have to provide the host IDs for every sample. So in this case, it's just assumed that every sample is a separate host. But if you have multiple samples per host, you have, of course, also have to provide for every sample in which host, from which host they came. And then if you ask what they are, this object has, and then you see that there are sequences, the sampling times, and the hosts are also in this Nubel data objects. So then from these data, you can enter the settings for the analysis. So it's mainly the priors by calling the function Phybreak, and you give the data set, and you can give a lot of information. So now I don't give so much, but you can give a lot of information about the priors. And whether you want to estimate the generation intervals or whether you want to have them fixed. So in this case, I provide already a mean sampling interval and the shape of the sampling interval distribution. And I make the prior for the sampling interval mean a little bit more narrow than the default, where it's very wide. And then what you see is that this results in a Phybreak object with 0 posterior samples, because we haven't done any sampling yet, and it shows you what the data are and what the model is. So that the generation, for instance, the generation interval, the current state is that it is gamma distributed with a shape parameter of 3 and a mean of 1, and that once we start doing the MCMC updating, the prior of this mean will have a mean of 1 and an infinitely large standard deviation. So it will be very uninformative. And you see that the prior of the sampling interval is as we entered in the line at the top of this slide. So then we can do the burn-in, where you have to give the number of cycles, and each cycle is in fact an update of every. So every host is a focal host once, and every parameter is updated once, and that is what is a single cycle. So in this case, I did 5,000 cycles, and what R then does is every 10 seconds by default, it gives you the state of this procedure, how far it already is. So you can get an indication of how long you still have to wait. And it gives you some of the parameters, the current states of the parameters, and the current parsimony score of the phylogenetic tree. And that also gives you an indication of whether you are, it is approaching some kind of equilibrium, or the, how is it called? Whether it has converged. Because in this case, the data set contains 26 SNPs, so the most parsimonious phylogenetic tree also has a parsimony score of 26. You see that already very quickly this score is reached, which indicates that there's already convergence very quickly. But sometimes you see that this takes a little bit more time to get a proper tree with a low parsimony score. Then after the burn-in, you can do the sampling, and then all the samples will be stored in the Phybreak object. So once we've done this, you see that we have a Phybreak object with 25,000 samples, and of course the same data and the same model. And then the package contains some functions to explore this, analyze this output. So first of all, if we attach the CODA package, where you can analyze your MCMC, just the basic MCMC and diagnostics, you can make an MCMC object with a built-in function, and for instance plot the trace. So this is just an example that I did for the log likelihood. You can use all the CODA functionality. And there is a function ESS, which gives you the effective sample size for all the parameters and variables in the model, so that you can also get an idea of whether you already have enough samples. And there is a function to get summary parameters or summary statistics for the estimated parameters. But as Caroline also told you about for Transphilo last time, that those parameters are not of prime interest. They are mainly to check maybe whether they are realistic. But the main interest of the output is the transmission tree and the phylogenetic tree. So you can get the maximum plate credibility phylogenetic tree from all the samples with the function philo tree, and you can plot this tree. And then automatically the tree is colored. So this is the coloring which indicates in which is the coloring that shows you the transmission relation between the samples, which is also the transmission tree. But this is not necessarily the best consensus transmission tree, because this is really the the maximum credibility tree, so it's the best phylogenetic tree, but that doesn't necessarily also be the best consensus transmission tree. So there is another function, trends tree, which gives you the consensus transmission tree, and there are a couple of algorithms implemented, a couple of different methods to get a consensus transmission tree. And for me, I think the best is the Edmonds algorithm, which just for every case, by default, it would select the infector as the infector that is sampled most frequently in the MCMC. But if there are cycles because it can be that two cases have each other as most frequently sampled infector, then this Edmonds method will resolve these cycles by kind of second best choice. And you can plot this consensus transmission tree in this example plot. And this plot shows you, so on all the lines are the hosts in the data set. The crosses are the times of sampling. All these gray blobs are the more or less the infectivity curve. So it's the generation interval distributions. And you see the arrows depicting who infected whom. And the color indicates the support of those transmission links. And in this result table, you also see the support of those transmission links. Actually, for this data set, many supports are very low, and that's because some of those samples, there were no sequences available for some of those samples. So actually, it's not a very good example of, it doesn't really show that the methods really can work very well in some, if the data contain a lot of information, because this data set actually does not contain so much information. And then so this is not yet on the CRAN version, but in the newer version on GitHub, I've also made the possibility to plot the transmission tree with all the phylogenetic mini trees in the individual hosts. So this is basically the same, it's a different consensus tree, so not Edmonds, but a different method. And you can also see all the mini trees within the individual hosts. That's really a complete output. So then finally, a little bit about some new developments that I'm working on, or that I have already developed. So first of all, I've already promised to say something about this within host model. So in the CRAN version and the published version, there is a complete bottleneck at transmission and only one sample per patient. But on the GitHub version, you can have multiple samples per host. So that means that you get more tips in the within host mini trees. And you can have a wide bottleneck at transmission, which means that more lineages go from each host to the infector, which, as a matter of fact, also increases in more tips in that infector again. So you get really much more complicated within host mini trees with these extensions. And here you see, so this is just a simulated small outbreak where with these extensions in it. So you see in every host 3 sample tips, and you see, for instance, from host 5, you see 2 lineages in the transmission bottleneck going to host 3. Well, as a result of this extension, well, as I explained in the MCMC updating, all these mini trees are simulated. And by completely simulating, letting everything free in these simulations, you get so many possibilities for these within host mini trees that I, in first instance, saw that many proposals were rejected because most of the mini tree proposals were just worse than the than the current state of the tree. So in these, so I had to change, put more restrictions on those simulations of the within host mini trees. But now, in many cases, it works pretty well. So that's something that I'm still working on writing a manuscript about. And in the new version, I've added the possibility to give locations of farms or just a distance matrix. So it can be spatial distances, but it can also be other types of distances that can be used as part of the likelihood to give extra information, prior information of more possible or less possible transmission links. So that's some of it already implemented, some of it's still work in progress. So now summarizing, I've explained to you something about the outbreak analysis with Phybreak, where we can find out who infected who and when in densely sampled outbreaks. But in this package, the assumption is made that all cases are observed, or at least most cases. But in fact, basically all cases are observed, and the observation that's done is the sampling time and the genomic data. So what the package does is simultaneous inference of the phylogenetic and transmission trees to infer the transmission history, accounting for the complete uncertainty in phylogeny. I've explained to you the models for transmission sampling within host diversity and mutation, and especially this coalescent model for the in-host phylogenetic mini-trees. And I've demonstrated a little bit about, I've shown you a little bit about the MCMC sampling procedure, and shown you some methods to analyze the output, including these consensus transmission trees and these plotting functions. And finally, I've told you a little bit about further extensions that have been implemented or that I'm working on, such as the multiple samples, wide transmission bottlenecks, and use of spatial data. So finally, I would like to thank Caroline, not only for suggesting me to give this lecture, but also for helping me discussing and helping me work on this package, as well as Xavier Didelot and Jantien Bakker and Jarko Wallinga, two colleagues from RFVM who also worked with it. If you want more information, apart from the paper in PLOS Computational Biology, there is this Handbook of Infectious Disease Data Analysis, where I wrote a chapter on outbreak analysis using TransPhylo and Phybreak as examples. And you can go to my GitHub page and send me emails, of course, with questions. Thank you.
All right. Thank you very much. That was very clear and very interesting. I'm impressed that you sort of have made this like pretty complex MCMC procedure with phylogenetic likelihoods all sitting inside of R.
Yeah.
Did you write all of that? Like you wrote the Feldenstein pruning algorithm all like on your own, basically?
Yes. Yeah, I did. Yes. Yeah. So the idea was derived from this paper in genetics by Rolf Ipma, but yeah, basically I wrote it all in R. I did that myself. Yes.
Yeah. So I mean, I guess even a little bit farther. I mean, you've given some sort of future directions, but I mean, even a little bit farther. I mean, how far do you want to push this? I mean, I'm not an epidemiologist. You come into contact with epidemiologists a lot, and I mean, they do have relatively rich data, right? I mean, are you going to keep on trying to like add increasingly complex types of data that allow you to sort of bring that epi data and the genetic data together?
Yeah, so for me, I think most promising is to include more epi data, because when I present this to epidemiologists, to people really collecting these type of data, what they often want is so put more constraints on who infecting whom because they knew they were in a particular room or they were not, so I think adding more complexity to the genomic model, I think that's not directly the. So I've now included this wide bottleneck, but I think I don't want to go further there. I think for me, really the next step is try to include more epidemiological information, even so that you could include cases. So now if I have cases for whom I do not have sequence data, I only have the sampling time, and that just creates a lot of uncertainty. But if you would have epidemiological data for those cases, then it may even be possible. That's just, maybe it's just fantasy, but to have data sets where only part is sampled and sequenced, but for part of it there's only epidemiological data available.
Right.
But some data that do give information, of course, about likely transmission routes. That's really the way forward that I'm thinking.
Yeah, I mean that's. I mean, I guess fossils and sort of hidden data is always sort of a bit of a challenge for phylogenetic methods in general.
Fossils, yeah. Fossils.
Well, I just mean just in general, sort of like these unsampled things that we don't know about.
Yes.
I should remind people. Yeah, feel free to. I do have to wrap up in about 5 minutes, but if you can send me a tweet on Phyloseminar or post a comment on the YouTube.
So one of the things, if are there any questions? So
yeah, I haven't seen any yet, but I'll bring them up if they show up.
Because so one of the things that Caroline explained what TransPhylo can do is deal with these unobserved cases.
Mm-hmm.
So she had this probability of actually observing a case. So if you, so that some cases are not in the data sets, but that you can take account of those. But I've seen, so I can, that is actually very problematic if you combine that with the wide bottleneck.
Mm.
Because you can see here in this figure, in this host 5, you see that from host 9 to host 5, there goes a lineage, and then it goes on to host 3. But there are no samples in host 5 that relate the samples in host 9. So there's, in fact, the genetic information that will come from host 9 and host 5 doesn't give any information about the fact that 9 may be infected by 5. So if you also include the possibility that there are unobserved hosts available, then it can be, so now these lineages go through host 5, but it can go through any host, basically, and also through any unobserved host. So that's one of the problems of this wide bottleneck assumption. It creates a lot of uncertainty. That's what I've observed. So you must be very sure that it is really occurring before choosing this option in your analysis.
And so to be clear, what that would, I mean, an example of that sort of thing is where you feel confident that multiple strains are being passed from infector to infectee.
Yeah, so if you are sure that that occurs, then it's only useful if you also have many samples in each host, so that it's likely that there are not so many of those lineages going through hosts without observation, like here in host 5. If there are many of those instances occurring, then it will be almost impossible to infer who infected whom. Yeah.
Yeah, I mean, it seems like that would also make the MCMC sampling a lot slower, or maybe your acceptance rate to be.
Yes, exactly. Yeah, that's what I'm observing. Yeah.
So I mean, I guess in general, I would imagine that your acceptance rate is sort of like it's. I mean, as these things get big, I would imagine the acceptance rate does go down quickly.
Yeah, so the acceptance rate goes down if there are many samples within hosts, because then there are so many possible mini trees in each update that many of those are just not very likely. And also if the genetic data are very rich, so if there are many SNPs, because so it goes quickly if there are many if many of the within host mini tree proposals are more or less equally likely, so that it doesn't really matter a lot. But if they are very rigid, these within those mini trees, then updating goes very slowly. So then maybe I would think that if the genetic data is so rich, then basically the phylogenetic tree is more or less, there's not so much uncertainty about the phylogenetic tree. So then I would think that this transphylo would be a better way to.
So I mean,
to analyze your.
I mean, I know this is a phylogenetic seminar, but I mean, how important are the mini trees in a sense? Like, what if you just took sort of a star tree model and you still had sort of an ancestral population size or something like that? You still had sort of a coalescent model, but well, I guess it wouldn't be really a coalescent model, but is there a sense in which you could simplify things by not thinking about the trees explicitly within the host?
Well, I'm not sure what you would then suggest. Yeah, I have been thinking about this to resolve, but I'm not sure if we are thinking the same thing then. So one thing, of course, is just assume that there is mutation at transmission, but I don't think you mean that.
I was thinking a star tree where you don't assume, I mean, I guess this wouldn't be a coalescent model, but where you just assume that each lineage inside an individual is evolving independently from the others. So it's just a tree where they all have a single common ancestor.
Okay. But then, I haven't thought about that option. I have thought about the option to, so for every nucleotide to assume that that there is just a number of variants present within the host. So basically for most nucleotides, there will always be one variant present, but that every nucleotide more or less evolves independently. So then you don't have a phylogenetic tree anymore.
Anyway, just a random suggestion. I do need to go now, but thank you very much, Don, for a thought-provoking and very timely description.
Well, thanks. I'm looking forward to the next.
Yeah, we'll wrap up, and the next one will be Eben Kina.
Yeah.
Very much.
Okay. Thank you.